Well, hello, everybody. Good morning, good evening, good day, everybody, wherever you are in the world. Welcome to the Global Ocean Observing System Biology and Ecosystems Essential Ocean Variable webinar series. Today, we're going to talk about phytoplankton and zooplankton biomass and diversity. These are two of the things that we call essential ocean variables. And so we're going to go over that. This series of talks is a collaboration between the Global Ocean Observing System, the Marine Biodiversity Observation Network, the Air Center's Networking Fridays, and the European Union project called BioEco-Ocean. And I think if somebody can paste the link to these series of talks in the chat, I would welcome that, so that everybody, there's a series of talks that already happened about other EOVs, and there's some more coming. I'm Frank Miller-Carger from the University of South Florida in the United States, and I will be your moderator today. I'll try to keep your session on time and pass your questions to the speakers. As you hear the speakers, please use the question and answer interface and at the bottom of your Zoom screen to interface and pose your questions. So today we're honored to have 4 speakers who are members of the Biology and Ecosystems panel of the Global Ocean Observing System. There's 3 panels. There's one on physics, there's one on biogeochemistry, and then there's the biology and ecosystem. They work together in trying to see how we measure the ocean. First, we will have Rafe Kudela. Rafe is a distinguished professor in the Ocean Sciences Department at the University of California, Santa Cruz. He's an aquatic ecologist. While he works a lot on marine systems, he also has done a lot of work on the land-sea interface in freshwater systems. He has conducted extensive research on the ecology of phytoplankton and has focused a lot of his time on harmful algae blooms. Kudela is past chair of the Global Ecology and Oceanography of Harmful Algae Blooms program of the IOC, the Intergovernmental Oceanographic Commission, and SCORE, and was co-chair of the U.S. National Harmful Algae Bloom Committee, and is a member of the U.S. National HAB Observing Network. Claire Davies will go next. Claire works at CSIRO in Australia, and she comes to oceanography after an initial career as an engineer in the oil and gas industry. It's an amazing jump. She is the project leader for biogeochemistry at the Integrated Marine Observing System, IMOS, which is Australia's contributions to the GOOS. Claire leads the IMOS Biological Ocean Observer Development Program, and is an expert in phytoplankton and zooplankton, including taxonomy, is an expert in data management and analysis. Then we will go to zooplankton. Svenja Halfter is a zooplankton ecologist and biogeochemist working at the Earth Sciences in New Zealand. She focuses on the ocean carbon cycle and climate change impacts on zooplankton and vice versa. Svenja works in the Southwest Pacific, including the Southern Ocean and Antarctica. Then Kim Bernard is a professor of biological oceanography at the College of Earth, Ocean and Atmospheric Sciences at Oregon State University in the USA. She's a zooplankton ecologist who focuses on krill and the role of zooplankton in marine ecosystems. And her work spans the Southern Ocean, the Bering Sea, New Zealand waters, and the Northeast Pacific region of the state of Oregon in the United States. Kim has worked at the Palmer Antarctica LTER program and helped establish South Africa's first coastal long-term monitoring station. So before I give them the floor, if I can have the next slide, please. I wanted to go over this diagram that is a representation of what we call the framework for ocean observing. That was, it's a pretty much common sense approach that the Global Ocean Observing System came up to try to understand what is it that we need to measure in the ocean. So I'm going to start at the left-hand side with the requirements for international, local, and national things that we have to measure. GOOS uses these panels, the physics and climate, the biogeochemistry, and the biology and ecosystems panels, to identify these requirements and capture them in what we call essential ocean variables. These are the ones that define the kinds of observations that we have to collect, and in particular, we're interested in biodiversity and biology observations in what we call the essential ocean variables. So these are, and the speakers will go over this, there's a number of things that end up defining what an EOV is all about. And we use the networks like the Marine Biodiversity Observation Network and many others to try to get the community together on how we organize to collect these observations in a way that is standardized and then pass them on to databases like OBIS, the Ocean Biodiversity Information System, and another metadata database that is called ODIS. These are managed by the Intergovernmental Oceanographic Commission. There are other databases, and the point is to try to put into these things that we consider necessary for operations, so operational oceanography, for management, for research, in a way that we can do large-scale research and understand the large-scale impacts on local ecosystems. These observations are used by other groups, by many of you, and you assemble them into maps and time series in what we call essential biodiversity variables. So these maps are essentially aggregations, computer models, and in some way or other synthesis of these essential ocean variables. And then those are picked up by the various organizations internationally, nationally, that try to do assessments, looking at sustainable development goals or weather impacts or food and so on. And then those groups provide feedback to GOOS on what could be changed. So I hope that more or less that gives a sense of the relationship between essential ocean variables and essential biodiversity variables and indicators. All of these three things are pretty well connected. And I will let Rafe go next and explain specifically the phytoplankton EBVs, Rafe and Claire.
Thank you. We switched to Claire going, so I'll let her start.
Hello, everybody. I'm Claire Davis. Thank you very much for joining and being interested in the EOV process that we're going to talk to you about today. Next slide, please, Svenja. So I guess the first question is, why phytoplankton? Why do we want to measure phytoplankton? So it has a lot of very important roles in the ecosystem. So it produces oxygen, obviously very important. It's also very critical to biogeochemical cycles, as you can see in the image on the screen. Plays a very important role in exporting carbon down to the depths of the ocean. It's also a very good indicator of environmental change. So the processes within phytoplankton are very influenced strongly from temperature. And this means that fast, rapid response, when you can measure that, is a really good indicator of what's going on in the water. It also impacts water quality by the nature of what taxa is present and in what abundance. And that leads into the formation of blooms. Now, these can be really positive, highlighting areas of high productivity and feeding hotspots for higher trophic levels, but they can also be harmful as resulting in things, mass mortality events and deoxygenation. So there's very many reasons why we measure phytoplankton. And there are just as many standard ways and accepted methods of doing that. So in the specification sheets that we're using to define the EOVs, we're not trying to be prescriptive about what you must do. We're trying to provide a range of options that you can use that are scalable from individuals with minimum resources up to best practice, large-scale, long-term programs. So we're trying to give you options, we're trying to give you advice, and we're trying to help how you can structure your observation programs. Next slide, please. So what do you measure? So there's a lot of questions around what and how to measure phytoplankton. And most of that will depend on your question and your resources. So again, in the spec sheet, we're trying to define what is required at a minimum level, but then we can expand that up to what would be useful for best practice and how we can get there. So we can do it in a stepwise way. So there's various scenarios that we can answer for various questions depending on what you're interested in the environment. So you might be interested in long-term change, or you might be interested in the role of phytoplankton in nutrient recycling, for example. So as well as the information in the sheets that's there, we also provide a lot of links to standard practices and methods. And there's more information that you can follow from the sheets if you follow the links. When we look precisely at what to measure, what you see on the slide here, it's not exhaustive. It's just an example that hopefully gives you a starting point of where to think of how to go forward with measurements. So the actual EOV itself is defined as phytoplankton biomass and diversity. The biomass typically refers to either the mass as a concentration per unit volume or the abundance and quantity of organisms in a unit volume. And the ways, for example, that you could measure this include collecting water samples that you can then use to measure pigments with methods like HPLC, or perhaps flow cytometry to look at the phytoplankton, or you could look through satellite remote sensing techniques at more bulk chlorophyll, and in some cases functional groups are becoming able through those techniques as well now. The phytoplankton diversity or composition refers mostly to the variability among phytoplankton. And this could be genetic diversity, taxonomic diversity, even size class diversity. And the way you would measure composition, traditionally that's been mostly done in water samples through microscopy. And there's also now coming into the becoming popular things like imagery techniques, so using instruments like FlowCam. There's also a lot of molecular methods now, so through water filtration and collecting samples for eDNA. So once you've collected your prime phytoplankton data and observations, you're going to need to interpret that. You're going to need to provide some context to those observations. And again, the way you can do this is with supporting variables. And these are numerous and also depend on your question and resources. So in the spec sheets, what we've tried to do is suggest some supporting variables that you may want to use to answer your questions. And we've tried to specify whether they would be absolutely required variables, or perhaps they're less important and we would class them as desirable. You know, there's a lot of cases for everybody wanting to measure everything in the water, and you know, the resources often preclude us from doing that. So as well as the supporting variables that I've got listed there, you can also use sort of biooptical variables from remote sensing, and maybe some physical characteristics like mixed layer depth or surface currents. Next slide, please. So as you can see from the way I've been talking, there's quite a lot of challenges just to making the measurements and putting them in the right context, and feeling that what you're doing is useful. There are a lot of sampling methods out there, and they all give varying degrees of information. There's older methods like microscopy, and then there's the newer methods like the omics techniques that are coming in, and they all require very different very difficult statistical techniques to use the data. They show very different volumes, and they come in very different ways of using them. So there's a lot of questions as well within, like, do you start something new? Do you carry on something old? And also a note there that within the bioeco context, phytoplankton overlaps with other EOVs, and we do tend to include cyanobacteria in with phytoplankton as well, although you can also find that in the microbes EOV as well. Next slide, please. So this is an example page from the specification sheet, and in this slide we're looking at the phenomena that we've chosen to observe and to give you information on in more detail through the specification sheet. Again, this isn't exhaustive. We've picked looking at stages and trends, role in transport and cycling of elements, and hab occurrences. But there's lots of other examples and reasons why you would study phytoplankton. But what we're trying to show here is a structural method in which you can use your observations to plan and implement your research. So we try and give information that can help you go from minimum, like a small scale, so local, up to global, and help you understand that if you're looking at a long-term monitoring project, then you may not need as much temporal resolution as you would if you were doing a hab occurrence, when you would want to monitor a lot more frequently, but possibly overall for a shorter time. So we try and give a lot of these details in the specification sheets so that you can plan around where your research is going. So following now, we've got some case studies coming up that in different ways will put into context the information and how we can actually start to monitor some of these things in reality and what's actually been happening across the phytoplankton EOV. Next page, please, Svenja. So the first one case study that I want to highlight is the Australian Integrated Marine Observing System. So IMOS is a regional alliance of GOOS. It's a government-funded long-term monitoring survey, which we are really, really lucky to have in Australia. And straight up, we admit straight away that this is out of reach of a lot of individual and smaller research organizations, but just to focus on what can be done and the impacts that you can gain from some of these large-scale, well-funded programs. So we've been monitoring plankton in Australia since 2008. So we have a 15-16 year time series now. The red dots on the map, they're the national reference stations. This is monthly sampling occurring cross-institutionally on a monthly basis. All the samples are sent to a central analysis lab, and the data from there goes straight to the AODN, which is the Australian data portal. And from that, it goes through to OBIS. So that's actually a requirement of the IMOS funding. And I think this is really important to stress, is if you want money from IMOS, you have to make your data open access. So for our terms, 6 months after collecting the sample, that data must be on the IMOS portal for everybody to use. So in terms of phytoplankton, we collect composition from microscopy samples. We collect pigments and flow cytometry samples, and we also do molecular work on Stereovex filters and for eDNA. And there underneath as well, you can see a list of the subvariables that we also collect to add context to this data. The continuous plankton recorder is the colored lines. Those are our transects. This is more like quarterly sampling across each of those different colored regions. Again, we have a central analysis lab, and again, that data must flow through to the AODN within 6 months, and from the AODN, it goes through to OBIS. So the impact of this data, we've been able to show an overall decline in phytoplankton abundance across Australia in that time period, and we've been able to confirm this with satellite observations. We have a direct route to policy uptake through the State of the Environment reporting. So the State of the Environment reporting is a report that scientists put together every 5 years, soon to be every 2 years in Australia, and the experts write about their area of expertise and how the long-term trend is, what the long-term trend is in that area. And all this information is collated into the State of Environment report, and that goes to the government. So that has a real big impact on policy uptake. The national reference stations have got over 400 publications, and for the ones that focus around phytoplankton, they're mostly around water quality, climate action, biodiversity, and food security. So it covers quite a wide range of impact areas. And we have also published our methods for the plankton sampling in Frontiers as a methods article, and that's for the first step towards best practice. Next slide, please. And from the large long-term monitoring, I wanted to then highlight the impact small data sets can have when they're collated. You hear us a lot talking about putting data into OBIS, and you might wonder what the advantages of doing that are. Why would you go to the trouble of doing that? But I want to highlight with this project how important that can be. So in Australia, we collated and standardized all the phytoplankton records in Australia. So these, it consisted of some really small local one-off studies. Some of them were student master's theses, right along to voyages and long-term monitoring projects. But you can see how many contributions we had by the length of the legend, and each legend isn't representing a study, it's representing a contributor, and so there could be more than one study within there. Some of this information hadn't been previously digitized, so some of the people who'd been working in the area for longer times still had information in notebooks that hadn't even been digitized. So we digitized that. And due to the fact that a lot of these came from different surveys, from different methods, from using different techniques, we've actually provided a really comprehensive metadata record with this data. And again, this is open access through the AODN and OBIS. And to get people to help us to do this, the carrot was that we published this data as a data collection in Nature Scientific Data. And some of the impacts that we were able to show from this, we documented quite a few range changes. One of the most notable ones was Noctiluca scintillans from the first record in 1860 on the east coast of Australia. We've tracked that range change all the way across the south coast, up the west coast, and also down into the Southern Ocean. We've been able to determine that some tripos species are really good identifiers of indicators of water masses, specifically a lot of the shade flora in the upwelling regions. We've got a lot better understanding of the dynamics of some harmful algal species in New South Wales. We've been able to use a lot of the information that we gathered on trichodesmium to help verify a biogeochemical model in the Great Barrier Reef. We've got a lot more species distribution maps with a lot more certainty around them now, and we've been able to understand the thermal tolerances of a lot of phytoplankton species in our region. And from this, we were also able to show in Port Hacking, which is one of the national reference stations on the East Coast, because we had data going back to the 1930s, we've been able to document a species that's got a preference for warmer water, replacing a species that has got a preference for much colder water. So if we put all those small data sets together, big things can come from those. So on that, Svenja, I'll pass over to Ray for the for the next two case studies, and thank you, everybody.
All right, thank you, Claire. And so thinking back to the phenomenon that we identified as examples of what we want to observe, we were just talking about status and trends, but another one was the occurrence of harmful algal blooms or harmful algal events. And so an example of if you actually put your data into these publicly available databases, being able to do more than you can with your own data set is this really nice analysis that Gustav Hallegraff led with many co-authors. And their fundamental question was, do we see more harmful algal blooms through time linked to climate change? And you can't do that at the regional level because regions may or may not track global patterns. And so what they did is they combined a couple of databases. And so they used OBIS to get the phytoplankton abundance, which is, of course, our core variable. And then they used another database called HADAT, which is the harmful algae event database, which is run by IOC, where member countries from all over the world submit records of when there's been a harmful algal event in their waters. And by combining those two databases, they were able to analyze these really large data sets globally and look at, are there actually trends that are statistically relevant across both regions and globally? And so I picked out one example from the paper where they've taken the data from those two data sets, and they show that yes, indeed, in Europe, there has been a steady increase in some of these harmful species, the data coming from OBIS. And using the HADAT data set, they can show that that steady increase is matched by a steady increase in impacts to humans. And then the far right panel shows that one of those impacts is they can see an increase in human poisonings caused by these sorts of events. And so we can go from just simply collecting phytoplankton all the way through to societal impacts by merging these data sets and using them in a combined fashion. And of course, what that requires is standardization. And so we need to know that the data sets are actually internally consistent, and they need to be accessible. And so those are the two keys. But what you get out of that is you can start doing more than you can do on your own. And so if you're interested, one of the big findings from this paper is they said, no, globally, there is no consistent increase in harmful algal blooms or harmful algal bloom events, but regionally, there are regions where it's occurring. And so again, the importance of understanding your own system and whether or not you're tracking a regional trend or a global trend. And so a very nice example of the data is available. You can bring experts together, mine that data consistently, and come up with a product that you didn't have before. Next slide, please. And so my second case study, kind of the opposite. And so HADAT is really just, did something happen? And the OBIS data was pretty traditional. We can understand the observation of phytoplankton numbers using microscopy or whatever else you want. But we're moving into an age where we're dealing with potentially really large data sets. And we know that one of the barriers is you say, well, I'd be willing to share my data, but it's difficult. It doesn't follow the Darwin core conventions. It's difficult to get into OBIS, et cetera. And so I want to give you two examples of groups that have worked around that or worked with data producers. And so on the right, we have something called the pelagic size structure database. And this is a curated database that they've taken the size of particles, primarily phytoplankton and zooplankton, from various instruments. And so across the top, you can see they're using imaging flow cytobots, ZooScan, UVPs, other types of instruments. There's FlowCam data in there as well. And they've developed their own data set that is, again, internally consistent, that has consistent metadata. And so that gets to one of the phenomena we want to observe. If you're interested in biogeochemical cycling and carbon fluxes, a big component of that is particle size distributions. And so depending on what data you work with, 100,000 data points may not be particularly large, but having it all in one place and having it consistent with pretty good global coverage is a huge step forward. And so this is available publicly, and they update it approximately annually with new data. And so there's QA/QC that goes into that to make sure that when you use the data, again, it's consistent, it's following standards, it's well documented. The other one, of course, is the rise of imaging flow cytobots and tools like that. And so they produce massive quantities of data. And so just as an example, Woods Hole Oceanographic Institution, where the imaging flow cytobots were developed, they have public libraries of millions of images that you can access. And so an obvious question is, well, what do I do with 1 million images? How do I handle that? And so as a community, we've been working on tools to make that easier to deal with. And so the little bit of text you can see on the left, this is a project that we were involved in where we developed a pipeline where you can take the imaging flow cytobot data and provide a series of tools that are available on GitHub to move it from data coming off the instrument all the way through to Darwin core compliant imagery that can go into OBIS. And OBIS does accept imagery that humans have not looked at. And so there is a metadata tag that tells you this phytoplankton image was identified by a human, or this phytoplankton image was identified by some sort of machine learning tool. And so we're trying to automate that. We're trying to get people to share their data and to follow Darwin Core standards. And that's where we're getting into really, really large data sets. And so there's a group that has started relatively recently to pull that imaging data together. And they've already got over 1 billion images, and they're aiming for 3 billion images. And the goal of that is to develop something called a foundation model, where they would be able to build a universal model that can identify plankton images and then could be used for whatever science that you want to do. And so again, the advantage of participating and saying, well, yes, it's kind of difficult to get my data in there, but once it's in there, you can start sharing these tools, you can start sharing data analysis, you can follow best practices and know that what you're doing at your site is consistent with what people are doing in other sites. And so that's really one of the big focuses of the BioEco panel and the EOVs is, again, not to be prescriptive to tell you that you have to do this, but to show you what the benefits are. What do you get by sharing your data and sharing your data consistently? And with that, I'll turn it over to Kim, and she will continue on with the zooplankton.
We also switched around, so it's me. Yeah, thanks for having me today. I will talk about the Zooplankton EOV. I'm part of a team with Kim, so yeah, happy to be here today. Of course, it stopped working now. Oh, here we go, do it myself. As you heard before from Frank, we of course focus here on zooplankton and the zooplankton EOV that we just updated this spec sheet. It's part of GOOS, but obviously there are plankton components in other variables across different initiatives. For example, we also have the Global Climate Observing System. They also have a ECV for plankton. And we are also part of GeoBond, so within community composition, ecosystem functioning, species population, species traits, we will find a plankton component in these ones too. Plankton and zooplankton in particular contributes towards sustainable development goals as well. So we are well connected. And you're just starting out with this, because I only started at GOOS a year ago, a bit over. It's very confusing because there's a lot of acronyms floating around, but we're getting there. Zooplankton is all drifting animal plankton, and people put here some errors, but you could argue that some of those I put the errors, and the lines are a bit blurry between groups. So some of those, for example, dinoflagellates, some people would argue they are actually phytoplankton. But because some of them are mixotrophs, so they eat and also photosynthesize, it's just getting very complicated. Whereas humans, and scientists in particular, we're trying to put things in different boxes, but in the end, what matters is that these are covered by ocean observing systems and monitoring methods that we use. So in the end, it doesn't really matter if we call it phytoplankton or zooplankton or just protoplankton or whatever you use, as long as we monitor the changes. And also there's likewise, on the higher size classes like salps and jellyfish and siphonophores, some people, and fish larvae, actually, they would be called micronectin by some people. Again, doesn't really matter which box we put that in, as long as it's covered. And zooplankton is very diverse, so it's not even just one group. It is, as you see, like ranges from so many different sizes and so many different groups, all drifting with the water currents. But what they have mostly in common is that they're quite important for the aquatic food webs. You can see here, this is very simplified, obviously, but it connects phytoplankton to higher trophic levels. And like the energy flows from phytoplankton through to zooplankton, usually to higher trophic levels. So they're quite important. Likewise, as already mentioned earlier, by feeding on phytoplankton and repackaging material in the water column and vertically migrating, they're injecting carbon as well. So zooplankton play a very important role in the nutrient cycle and carbon export and food webs in general. And because they're quite diverse, there's so many different monitoring methods. And as also said, there's some more historic methods like the continuous plankton recorder, CPR, and nets that have been around for over centuries. Whereas other things that were coming up recently, molecular data is getting more and more prominent with eDNA. In situ imaging is now on the rise too, which is very cool. But again, we're producing a lot of images, as just mentioned. How do we deal with that? There are pathways now, but we're only slowly getting there. There's moorings, there's acoustics. So, and then the challenge, of course, in the end is to integrate the different data streams. As I said, there's also the problem that zooplankton is patchily distributed in the water. So it's not only that it's just evenly you can sample and you get zooplankton. So mostly some species are because they're quite common. Others are more in swarms and you have to really, or like only up during the night, or so it's patchily distributed. Even if you do a back-to-back tow, there might with a net, there might be a completely different abundance you get. And because of its high diversity within zooplankton, there's no one-size-fits-all sampling method. Unfortunately, I wish there was. So every sampling method comes with biases. So in terms of species or size class related, it makes a difference if you have a 100 micrometer mesh or if you have a 300 micrometer mesh. You get a different community, and the challenge is then to understand what exactly is the real community out there, or maybe at least quantify the biases to get there eventually. And that's also the problem with data standardization. It is not straightforward because we use different sampling gear, but also the way we deploy things is different. So if you have published data, the raw data is important, of course, but not the raw data, but like the data itself, the numbers itself is important. But what is equally important, or even more important, is the metadata, because we are relying on understanding how the different gears deploy to understand why there's differences in the data sets, if you want to combine them all together. And then the next, oh yeah, Kim is taking over from here with some case studies.
Thanks, Svenja. Hi, everyone. Thanks for joining us today. So Svenja did a great job of explaining how complicated it is to monitor zooplankton. The standardizing, I think, is probably going to be the biggest issue. But when you can standardize collection methods and make the data readily available, there are many globally significant research questions that you could address. And the best case study for this is really the Continuous Plankton Recorder Survey. And that's already been presented a little bit already, because you can also collect phytoplankton samples with it. But the CPR survey has been ongoing since about the 1930s without any major change in design. So it hasn't changed over that time. And it's been amassing a huge amount of data over a very large area of the world's oceans. So because CPRs can be deployed on ships of opportunity, they really can go just about anywhere. Now, the collections are restricted to the surface, though. So for zooplankton that migrate through the water column, we're not going to get everything. We're really only getting those zooplankton at the surface. And you can see the little opening at the front end. Svenja, if you can use your mouse and just point to that, that would be great. It's pretty narrow. It's totally fine for phytoplankton. We're going to miss most of the macrozooplankton and, of course, the megazooplankton. So there are some limitations, but despite this, valuable zooplankton data have been amassed from the CPR program. And I'm just going to give you two quick examples on how some of these data have been used. So Svenja, if you want to go to the next slide, please. Okay, so the first example is this one. It's a paper led by Max Campbell, where they used the global CPR database of copepod species abundances to test Bergmann's rule. So essentially, the members of the same clade are larger at colder temperatures. That is Bergmann's rule. In addition to temperature, they also modeled body size of copepods against food availability using chlorophyll, and the proportion of omnivorous copepods in the community. And in their words, they found that as the world warms, smaller copepod species are likely to emerge as winners, potentially reducing rates of fisheries production and carbon sequestration. So as you can see, this is an incredibly powerful finding that has global implications. And it's essentially from this program that has been ongoing since the 30s. You can see the structure of the CPR. Nothing has changed. It's kind of amazing. Okay, so we can go to the next slide, please. All right, so this example is another one of how accessible, standardized zooplankton data can be used to address globally relevant questions. So here, Martin Edwards and his colleagues investigated changes in krill abundance in the North Atlantic over a 60-year period. And like the previous example, they also used CPR data. Now, while they found that a 50% decline in surface krill abundance occurred, they also found that this was not associated with a range shift. And instead, they described a habitat squeeze of about 4 degrees latitude that likely explained the decline in krill numbers. So these are two, I think, excellent examples of how a sustained standardized zooplankton observation program, the CPR survey, can allow us to address global phenomena of ecological and societal importance. But as fantastic as the CPR survey is, and it truly is, it is still just scratching the surface, literally. It is literally just towing the surface of the ocean. So zooplankton observations of a similar resolution and extent that extend into the water column would tell us about vertical range shifts, responses to shoaling oxygen minimum zones, the influence of global zooplankton on the biological carbon pump, and so on. So Svenja, yeah, you can change to the next slide, please. So this brings me to the GOOS BioEco Zooplankton EOV that Svenja and I are co-leading at the moment. And our aim is to establish a framework to standardize the collection of zooplankton biomass and diversity. So specifically just biomass and diversity. There are many other things about zooplankton that can be studied, but those are the variables that have been decided are the essential ocean variables for zooplankton. And so obviously to extend on what the CPR has already done and is currently doing. So collectively, the continued CPR survey and what we're able to do through the Zooplankton EOV could be used to detect a whole suite of phenomena, including trends in communities and populations, phenological changes, or biogeographic range shifts. So if you're involved in collection of zooplankton samples from anywhere in the world's oceans, you can contribute to this effort. We're in the process, as many of my fellow panel members have already said, we're in the process of updating the specification sheets. You saw the one, a sort of snippet from the one of the phytoplankton EOV that Claire shared a few slides back. There's a similar one for the zooplankton. And the aim is that this specification sheet will provide guidance, which is driven by a community of international experts. It's not just us, on standard approaches to collecting zooplankton data of relevance to the GOOS Zooplankton EOV. And then you can go to the next slide, please. And then on just one final note before we open for questions, OBIS has been brought up a couple of times through the presentation, but if you collect phytoplankton or zooplankton data, or both, that would fall within the realm of these two EOVs, then please consider archiving your data sets with OBIS. Importantly, OBIS accepts data that are compatible with Darwin Core. That term has been used already in the seminar, but essentially that is a standardized set of terms and guidelines designed to facilitate the sharing and integration of biodiversity information. So by using Darwin Core standards, you can ensure that your data are compatible with other data sets. You can promote interoperability and enhance the overall impact of your work. And if you scan those two QR codes, you can find out a bit more about OBIS and how to upload your data there. And yeah, that's all from me.
Well, thank you, everybody. Those are fantastic talks and great story came on what you can get from that device. It's amazing that it's so much coverage in the higher latitudes and nothing, basically. It didn't seem like there's even a single CPR trace in the tropics. There's a lot to do still, lots of research.
Yeah, they absolutely.
Yeah.
Yeah.
Well, I don't have any questions in the question and answer box, but if anybody wants to put one question in there or let us know if you have any questions, we would be more than welcome to try to answer them.
Should I unshare my screen?
Yes, please.
And I guess if nobody has questions to start, we could also maybe pose if anyone would like to answer this prompt. But if you were to use one of these data sets, one of these big global data sets, how might you use it? And is this something that you're interested in?
Well, there's a question from Goose on what is the best way for people to contribute. I think that over my career, I'll jump in here because I've dealt with this question. I know people that are very protective about the data because a student maybe developing a thesis or they're developing a paper. And my approach to this has been, I'll just get everything out there. We've managed a time series in the Caribbean called the Cariaco Ocean Time Series. And there I learned that the more we put the data out, the more people are interested in working with you. And the data gets picked up by anybody in the world, and they want to look at time series of biodiversity and phytoplankton. And you end up in more groups than sometimes more than you know what to do with. So it's a way to be open with your data. Does two things. One, if you integrate them into these databases, it allows you to see patterns at a larger scale that you would not otherwise see, and some of those patterns can help you explain your own data. So is it coming sideways through advection? Is it changing in synchrony, or is it completely disconnected and maybe driven by a local pressure? But it also allows you to participate in publications like Rafe mentioned that bring lots of people together to try to do a global synthesis or regional synthesis. So that's very rewarding. So that's one way that I've learned to work with people, is basically sharing my work and my students' work, and they get involved in any number of things. But maybe other people can have. So we're starting to have some questions in here. Dr. Hala Mohammed Taha, can we monitor phytoplankton or zooplankton without measuring physical or chemical parameters?
That's a
What?
should I can take a stab at that, at least to start.
Please.
I think that's a really good question, and I think we need to be careful about saying if you're going to measure, biomass of zooplankton, you also need to measure all these other things, because we want to try and get as much as we can. And I fully recognize that certain places might not have all the resources to measure all of the different parameters that might be necessary. I'm just going to say what I personally think, but I'd love it if anyone else chimes in. But I think the more we can get, the better. A lot of, most areas we'll be able to get, there's satellite imagery, sea surface temperature, ocean color for chlorophyll. So there should be options to gather some at least surface variables that will go alongside your zooplankton or phytoplankton data collection. And so I would say if you're able to collect anything, biomass and diversity or diversity of either of these, I think it's still valuable to share it. That's what I would say.
I'll chime in on things like the HADAT database, where it's just, was there an issue? You can put in that with no other information. And as Kim was saying, what we end up doing a lot is people have data on phytoplankton or zooplankton, but not all the other data we might want. There's often really good numerical models where we can get things like the physical parameters or the chemical parameters, but those models are generally not great at getting the biology. And so the biology that you're measuring is the most important contribution, because there's ways around getting some of that other physical and chemical data.
Yeah, and I would just point out that throughout this presentation, you've heard a lot about the continuous plankton recorder. The continuous plankton recorder only records phytoplankton and zooplankton and subjective color index from the color of the silk. They're towed behind mostly ships of opportunity. Very few of those tows happen on research vessels where there is ancillary data. So for the most part, none of that CPR data has any other data collected with it. There is a program to try and instrument the CPRs so that they can do this in a different way, but the way that the studies that Kim was showing and the work that we've done within the CPR program is mostly relying on satellite information for the and models for their extra parameters. So even a huge big survey like that doesn't necessarily measure anything other than just the plankton. So like Kim said, if you can measure something, measure it and share it. That adds to everybody's general knowledge.
I see. Sorry, there was a question from Antonella Desian. Sorry, I'm probably pronouncing your name incorrectly. I apologize about that. I accidentally hit the wrong button and it went into the answered section. So Frank, I don't know if you saw that or if anyone else saw that.
Yes, but you
But
can
they're,
answer.
yeah, okay. Oh, okay. Well, they're asking if it's possible to contribute to OBIS if they've already uploaded data in another platform. And I believe that it is totally fine to do that. But if anyone else wants to chime in, that sounds good.
Yeah, one issue is that at present these databases are somewhat independent, and eventually someday in the future, who knows, AI will solve all the problems of the world, and you may have all these databases linked together. And so if you try to do an analysis, so like some of the time series that all of you showed, you will fish the data from the different sources and develop an assessment. But right now, that is not a possible thing, and that's one of the goals of this system that the Intergovernmental Oceanographic Commission is developing, called ODIS, that tries to at least pinpoint where what data may be using metadata, but it doesn't link the data. So if you have the data in one place, like OBIS, then you can try to make relatively easy assessments. That OBIS provides some ready-made tools. You have an R interface where you can program your own way of mapping. But if you have to fish the data from many different databases, we all of us spend a lot of time doing that. That's part of our job and the way that we grew up. But eventually the idea is to try to make this easier, especially because we're trying to make these things operational and understand what is driving change. Okay, I don't see any other questions. Yeah, just sort of like this, there are, just like phytoplankton and zooplankton EOVs, there's we shared the link to the bioeco panel EOVs, but also biogeochemistry or and physics. There's lots and lots of things that we call EOVs, and what you measure, which of these you choose, is a challenge. Obviously, you can't measure everything everywhere all the time. So how do you select which variables to measure is definitely part of the challenge, and it depends on the problem and the question that you have. So Ashanti Canto Garcia asks, what remains a major challenge or gap that you would be able or to address in the future? So maybe in the last few minutes, and all of you could answer this challenging question. Claire, let's go with you.
Can you repeat the question again?
Yes, I think we lost Kim also. Ashanti asks, what remains a major challenge or gap that you would like to address in the future?
Oh gosh, everything.
Yeah, it's really hard to decide.
I think one of the big two questions that I would really love answered is how to get a better quantification from molecular data. So at the moment, it's sort of semi-quantitative. You can get relative proportions of different reads. What I would really like to see is that converted into an actual quantitative abundance, so it can be used in ways with other data a lot more easily. And as you highlighted, doing some bit more CPR work in the tropics would actually be really useful. We do actually have some data that hasn't made it through as far as has been analysed since we did the study with Max Campbell that Kim was showing. So there is some data in the tropics now, but it's still very, very far behind. So yeah, I think that would be really nice to see.
Yeah, I agree. There are some regions on the planet, that are undersampled because, I mean, North Atlantic, obviously, it's surrounded by a lot of countries that are focusing on research, have a lot of money, and to analyze things too, because there is also a lot of taxonomy still needed to analyze those CPR samples. And I mean, I'm just talking about New Zealand because we have one person to do that, and it's not heaps. So and we need to make sure that this knowledge is not dying either. So while everybody is now focusing on getting more molecular work out there, and it's really good because there's so many opportunities, but we need to still make sure there's some taxonomic expertise around to compare and validate our data. I think that is. And then undersampling of certain regions that are not sampled well yet. But also are impacted by changes, like all the whole ocean, the whole global ocean is impacted by climate change, but we have limited observations in certain parts.
Yeah, I would amplify that and say, one of the huge advantages of the CPR is it really hasn't changed, and so you've got, a 50+ year record where the method is identical, and that's really powerful. And so a challenge that we constantly face is, as new methods come along, how do you merge them? Because the longer the record, the more powerful it is. But if we switch to a new method and there hasn't been that validation that the molecular method is giving you something comparable to the CPR, now we've got two beautiful data sets, but you can't combine them. And so that's always a challenge. And I think it's something we can overcome, but we have to do it as a community to make sure that we agree to the standards, that we're comparing things as apples are apples and not apples or oranges.
One challenge that Dan Costa pointed out is by putting data in different places, if the data and the metadata is not complete, is that they may appear as duplicate or triplicate data sets, and sometimes they get counted multiple times or get averaged in a way that provides a bias. So that's an important point that Dan pointed out. And I also think that one of the biggest challenges that we have is how do we forecast life and biodiversity? Ultimately, everything that we do is how do we get to models and scenarios that can get us to predict the future of life. So anyway, we're at the end of the webinar. I want to thank the speakers, really fantastic talks, and thank you to the audience for being here and listening to these explanations about essential ocean variables. So thank you very much, and bye everybody.
Bye.